It is my pleasure and honor to introduce Silvio Tosatto. Silvio Tosatto, who is an Italian professor, was born in Bressanone. I'm quoting this. Why? Because he got completely his education abroad and at the University of Mannheim in Germany, where he got his PhD, and beforehand he got his also graduation. This is important. Why? Because he then came back finally to Italy within a project called Brain Gain. And this was in 2007. He was called back as an associate professor at the University of Padua, where in 2017, he was appointed full professor of biochemistry and bioinformatics, becoming one of the youngest professors in biochemistry in the Italian Academy. There, he's a very active group leader of a biocomputing group who got several achievements. First of all, Silvio, I should remember that is also the only Italian scientist who is a co-lead of one of the platforms of Elixir Europe, the most important platform for bioinformatics services in Europe, collecting 23 countries, and there he leads the data platform. He is also deputy of the Italian head of node, Professor Pesole. His interest covers several aspects of what is generally termed bioinformatics and computational biology, as you might know by now, where the main aim is to integrate the development of novel methods and their application to biological relevant issues. The group of Silvio Tosatto regularly participates in international benchmarking, like the critical assessment of techniques for protein structure prediction, CASP, the critical assessment of genome interpretation, CAGI, and the critical assessment of functional notation experiment, CAFA, where the tools are routinely very well performing. Major interests of the group are several protein function, protein aggregation, residue interaction networks, low complexity proteins, machine learning, personalized medicine, but mainly intrinsically disordered proteins, which I insist calling intrinsically flexible proteins, that have been the focus of several initiatives of Silvio Tosatto, and it's obviously the reason why he's so famous around. So Silvio, thank you for participating to this initiative of the Winter School, and I leave you the microphone.
Thank you, Rita, for the very nice introduction. And for those who don't know this, with Rita, we have a bit of an ongoing conversation about how to call intrinsically disordered proteins. And I also fully agree that the name is not probably not the best one, to put it mildly, but it's stuck in the literature. So I'm actually here to present something where I didn't coin the name. We were just picking up from what was already there. But anyhow, so today I will be talking about some of the things we have been doing regarding intrinsically disordered proteins, and of course, AlphaFold is the big novelty in structural bioinformatics. So I will be also telling you several things about how AlphaFold relates to this field. And let's start with an example. So the protein you see here is the cyclin-dependent kinase inhibitor 1B from human. Short name is CDKN1B, or alternatively also known as P27. This is a protein which is important as a tumor suppressor because it inhibits the cell cycle, and therefore is one of the ways the cell has to escape from tumor development. It binds to the cyclin-dependent kinase complex and is around 150 residues long. And what you see here is the AlphaFold prediction, as you can find them today in the UniProt page for this protein. So you see that this is the model confidence, where you have the high very high confident, low, and very low confident parts. But essentially, you see that this snapshot done by AlphaFold2 resembles pretty much what you would expect of cooked spaghetti in water, with some addition of some local structure here and there, which is being predicted. So the point here is what is going on with this protein. And this is in reality just one example of a much broader array of proteins which have been known for a while. And specifically for CDKN1B, this is a fully intrinsically disordered protein. Below here, you have the DISPROT evidence. DISPROT, as you will see, is a database for disordered proteins. It's manually curated, and it is backed up by literature evidence. So there were experiments carried out on this protein demonstrating that it is intrinsically disordered, and you have here different color codes depending on whether it is just disordered or it actually binds something among those regions. So this is just a very simple example to give you a flavor for what you can expect from the AlphaFold predictions. So intrinsically disordered proteins and regions have been around for about 2 decades. And for roughly 1 decade, they have been broadly accepted as a novel paradigm for protein structure, for this protein structure function relationship, or better, not a novel paradigm, rather an extension of the existing paradigm, because as you certainly know, the structure function paradigm has proven very powerful. We have a lot of protein structures, but there are these things which are dancing around. Here you have another example. This is a conformational ensemble, where you have a large portion of the protein which just floats around in solution. So the basic definition is that intrinsically disordered regions, IDRs, are protein segments that lack persistent secondary or tertiary structure. And this was very, very well represented in this editorial from a decade ago in Nature, where they said breaking the protein rules. And this discovery leads us to a reassessment of various aspects regarding protein structure and function. So coming back to AlphaFold, in the AlphaFold-DB paper published last summer in Nature, already there, the authors had a look at the human proteome and concluded that around 40% of the human proteome had low or very low AlphaFold prediction scores, so the PLDDT. And in the paper, they already relate this phenomenon, so this around 40%, to the presence of IDRs, which of course they didn't prove, but they used data from the experiments I'll be talking about later to show that these are strongly correlated. So one approximation is that whatever AlphaFold does not predict as properly folded, so these ribbons which are floating around the core of the protein, these could be IDRs. But what else do we know? Well, for one, IDRs are ubiquitous and functionally important. This is a previous study where it has been inferred that about 1/3 of the human proteome, that's 3 to 4 million residues out of 11 million residues in about 21,000 reference proteins. Well, that fraction is intrinsically disordered. And this has been linked along this size versus number continuum to the smaller end of the scale. So here you see 4 different scenarios. At the one end, you have the very large complexes, which typically are over 1,000 residues large. And these are very well structured, and you have beautiful crystal structures or cryo-EM structures for these. These are thought to be around 600 to 1,000 in the human cell. Then below this, you have the single globular domain, which is the mainstay of structural biology. And these are on average around 100 residues long, and they are thought to be around 35,000 different globular domains in the cell. However, below these, we have 2 different instances. One of them are binding motifs, is showing here that you have a globular domain which binds to a short stretch of another protein, which is thought to be intrinsically disordered. And here you have very few residues determining the binding affinity. Length about 10 residues for binding motifs, and they're thought to be around 100,000 in the human cell. even lower than this, there are post-translational modification sites, which are essentially just one residue, say a phosphorylation site, a serine or threonine residue, which gets modified and a phosphor group is attached. So the length in this case is one residue, and the estimated instances in the human cell are around 1 million. Now, the interesting thing here is that about 70% of experimentally validated post-translational modifications, or PTMs, are located in intrinsically disordered regions. And that most of these 100,000 transient conditional regulatory interaction modules are also located in intrinsically disordered regions. So there is a function for these 3 to 4 million residues you have here, and that function seems to be mainly related to shorter binding instances, the binding motifs, and the ability to modify residues within them. And you can guess that by combining these two functions, you can get a lot of signaling done in the cell without really changing much of the sequence. So the binding modes of disordered proteins, they have been described as sampling wide spectrum structure and dynamics. First of all, there's one estimate which places the accessible surface area per residue of an intrinsically disordered residue at about 99 square angstrom, whereas the typical globular amino acid has only about 58 angstrom square of accessible surface area per residue. So they are more widely exposed. Then here you can see a schematic of different types of behaviors for IDRs, which can go from the disordered and dynamic, where it just floats around like cooked spaghetti in water, disordered and compact, where you have something resembling a molten globule, disorder and extended, this may be a tail at the N or C terminus of the protein, or even a disordered region with residual structure. Down here, you see a better relationship between structure and dynamics. And again, you see that these can differ quite a lot from the disorder to disorder binding instances, which have been described. Then the so-called fuzzy interactions where the proteins bind. They bind more or less, the disordered part binds more or less on the same region of the protein, but the molecular details can vary. The context-dependent ones, which would be the ones where the same linear motif can change its shape. The polymorphic ones, where it just binds on the surface and there may be some differences, and the disorder to order transition, which is the classical instance of a linear motif. So in this case, you have this short piece of the otherwise disordered region, which binds on a globular surface and therefore gets structured. So to see a few biological examples for the various scenarios, here you can see P27 once again. So disorder to order transition. This is disordered on its own, and it binds on the surface of the CDK2 cyclin complex. Or you can have disorder to disorder interactions where you have, for instance, SIC1, which binds to CD4 in the SCF complex, and you see that it just essentially jumps around the structure of the CD4, but yet it maintains a region where it is more or less constantly bound. Another example would be P15 PAF, which is behaving like cooked spaghetti in water. And here, depending on the binding partner, and whether it's alone or there is a different molecule bound to it, you see that it behaves differently and it stays disordered, even in the bound state. So it jumps around, but still it's able to form an interaction. So with this, I hope to have convinced you that disorder is not just one phenomenon, it's actually a mixed bag of different things, which are, for the sake of simplicity, just put together with one simple name. What I didn't mention so far, because the molecular details are not well understood, is biological condensation or liquid-liquid phase separation, which is thought to follow exactly the same type of behavior. But again, the molecular details are not so clear, so I won't be going into that area. In terms of experimental IDP research, there are different experimental fields studying protein structure and function. And essentially, for intrinsically disordered proteins, you need a different set of experiments in order to describe them. Here, some of them are listed. Usually you don't have a nice crystal structure because that would obviously not be disordered unless it is missing a part of the residues. But again, that is not informative. So you would have to rely on slightly more indirect techniques ranging from NMR, which can still be pretty precise, to things like Sachs and Fret, and even things like circular dichroism, which just tell you about the percentage of secondary structure in your protein, if any, can be relevant. Then you have additional methods for detecting interactions. Again, these are pretty indirect. X-ray crystallography here can serve for the instances where you have disorder-to-order transitions, and functional assays, which again are pretty indirect. So experimental IDP research leverages similar techniques, but partially requires other techniques which were not so widely used for other types of proteins. And this has spawned a different field. So since Rita also mentioned that I'm involved in Elixir, within Elixir, so the European Infrastructure for Biological Data, there is a intrinsically disordered proteins community where I was one of the co-leads, but then that has been passed on to Damiano Piovesan from our lab, plus Wim Franken and Giuliana Dostagni, who are respectively in Brussels and in Budapest. And I should also acknowledge as former co-lead and as the driving force behind writing the white paper, Dr. Norman Davey, who's currently at the Institute for Cancer Research in London. So the community goals for the IDP community within Elixir are essentially to simplify IDP data access and dissemination, drive the curation of the IDP literature, develop a centralized knowledge base for IDP data. This is very important, as we will see in the following slides, to integrate this type of data into the Elixir core data resources because it was largely missing, and to improve in general tools for IDP analysis. But now let me start by giving you an example of a protein which we would need to analyze further. So this is the protein called Drosophila mastermind. And it is a protein expressed in the Drosophila brain. Now you can easily see that this is a very strange sequence. And you can see that here, for instance, there's a polyglutamine, here there's a polyasparagine, then there are some sorts of degenerated repeats over here. Some part of the structure seems pretty diverse. And then there's other bits and pieces, polyalanine, and you may wonder what the heck this protein does. And in fact, it is a protein which exists in Drosophila, and it carries out a function. So it is just an extreme case of something which developed along totally unforeseen lines, which took it away from the regular structure. So a first attempt to study this and similar proteins was done by Wooten in 1994, where he came up with a software which is called SEG. And SEG is widely used as a pre-filter in BLAST. So whenever you do a sequence database search, to mask out regions which are likely to produce many false positives. If you think about something like Drosophila mastermind, finding this region polyQ here, can give you a lot of other hits to proteins which can contain polyQ, but that doesn't mean that they carry out the same function, let alone that they are homologous. So in order to weed out these proteins, the program SEG, what it does, it calculates the Shannon entropy within a local sequence window. So it scans the local sequence, and it attaches a value to this, which is variable. So that typical globular proteins have high complexity values, whereas atypical regions have low values. For instance, polyQ would fall in this region. But interestingly, in the same paper, the author described an analysis of both the protein data bank and SwissProt. So SwissProt is the annotated part of UniProt, as you surely remember. And this is a scale for the sequence complexity on the x-axis versus the frequency with which it appeared. And let's first have a look at the protein data bank. So these, at that time, mid-1990s, were largely fully crystallized proteins, and the distribution you would get of those proteins follows more or less a bell curve. Now, if you take these protein sequences and you shuffle them randomly, you see that there is essentially no difference, and you get the same bell-shaped curve. So, in other words, the protein data bank, the part where we have structure, behaves with a roughly Gaussian distribution. Now, if you repeat the same analysis on SwissProt, which are known protein sequences, and you shuffle the DISPROT data set, you get the same result. So it's broadly speaking a bell-shaped curve. But if you look at the real sequence SwissProt data, you have this very long tail of low complexity regions. And what you find in here are precisely the type of regions you would find in Drosophila mastermind. So what you can conclude from this analysis is that there is a conservation for these long and low complexity regions, which disappears as soon as you start reshuffling them. So it is not just the fact that these are proteins which always are entirely comprised of polyglutamine, for instance. Rather, they have regions of low complexity, which are outside of other regions which behave in a normal way. And if you just reshuffle the whole data set, then this bias disappears. So what is the function, therefore, of these biased regions? And what are they for? This was one of the first attempts to characterize something which at that time was not yet known as intrinsically disordered proteins. Another attempt came almost 10 years later, and this was published by Burkhard Ross's group in 2002, where they had a look at the protein data bank, and they found these kind of cases you see here. So these are proteins where there is something more or less strange going on, ranging from a small loop here, which is connecting parts of the protein, and it's just either highly flexible, as Rita would say, or intrinsically disordered. Then you can have loopy ends at the end terminus here, for instance, which is quite long, and you see something. Or you can have wrapping loops here, a region where there's something apparently missing from the structure. Maybe it's binding partner, and therefore this is, again, very flexible. And then you can have entire loopy domains in the protein data bank, which are missing secondary structure. So the analysis they did was to try to relate these phenomena with the amino acid composition. And this is an enrichment plot. Then we can look at it from the bottom to the top. So starting down here, these are the loop regions in a subset of the protein data bank. And you can see that the loop regions are slightly depleted in hydrophobic residues, and they are enriched in some small and polar residues, and of course proline, which is well known to break secondary structure. If you take all the residues in the entire SwissProt data bank, well, you get a less marked distribution where everything seems to be around the average, with the exception of cysteine and to a lesser degree tryptophan. But cysteine is a very peculiar amino acid in proteins because it can form disulfide bridges. So if you look at what they call non-NORs, so the ones which have regular secondary structure, this distribution is virtually indistinguishable from all residues in SwissProt. However, if you look at the NOS distribution in the proteomes, so these would be the entirely loopy regions, you see that this is stronger even than the distribution in the loop subset of the protein data bank. So it seems to be a more extreme phenomenon where more hydrophobic residues are missing, and proline is even stronger as well as serine. So the definition for no regular secondary structure in their paper was more than 70 residues and less than 12% secondary structure. And the amino acids were sorted by flexibility from the most rigid to the most flexible, with a background as PDB. So disorder prediction since then has essentially turned into a subfield of machine learning, where many people and many groups have attempted to take a protein sequence as input, knowing that there is a skewed distribution of the sorted regions as compared to the average of the protein sequences, predict from the sequence the region which is thought to be disordered, like this region down here. So it's a very simple prediction problem for machine learning, and you can use all the techniques which are available in bioinformatics, most notably neural networks. But there are, of course, also other techniques you can use, and even biophysical methods, as we will see, work quite well. So prediction methods for disorder can be broadly grouped into 4 different categories, which pretty much resembles what you would get also for secondary structure prediction, for instance. You can have statistical methods, which are based on the amino acid propensity. Then you can also have physical chemical properties due to the fact that certain amino acids lack interactions. We will see an example for one of the methods combining these two, which is called IUPRED, in a few slides. Then one very remarkable feature of intrinsically disordered protein regions is that they have low evolutionary conservation. So you may be able to recognize them because they are the most variable part in sorry, a multiple sequence alignment. And at the end of the day, you can use artificial intelligence methods, so for instance, neural networks. Regarding the amino acid composition, here you have a different view, which came from one of our papers, and it is the enrichment or depletion of various types of amino acids on the subset of regions which are known to be disordered. And you can see that there is an enrichment at this end of the scale, which again are the most flexible ones, whereas the hydrophobic ones are depleted. So one of the hallmarks of disorder is few amino acids which are hydrophobic and many more which are polar or charged or proline or glycine. So then there is a compositional bias, and there can be different subtypes of disorder. This has been mostly studied by Rohit Papu's group in terms of charge separation. So they have been looking at regions of disorder where there is a high net charge and the charge patterning involving that. So by definition, you have low hydrophobicity. And in these cases, depending on the patterning of the charges, you can have different behavior of disordered regions of the same length. But in any case, they are highly disordered. And the regions with high net charge are quite common in the proteome. Finally, there's the conservation. This is a slide from a paper from 2011, where the group of Belay et al. Studied the conservation of disorder across multiple sequence alignments, and they came up with a distinction of 3 different types. You can have non-conserved disorder, which are the regions which are predicted to be disordered in your sequence, in your query sequence, but they are missing from other aligned sequences. Then you can have the so-called flexible disorder, where there is a piece of the sequence which is always present or many times present in homologous sequences, and it's always disordered. And then you have constrained disorder, which is a region which is strictly conserved across the different sequences you've been aligning to, and it's also always disordered. Out of these 3 types, the non-conserved and the flexible one are predominant. The constrained one is rather rare, but when you find it, it is telling you something about the protein function. So being on the lookout for lack of conservation is one of the techniques used by many predictors in order to establish that something is disordered. One method I would like to briefly mention is IUPRED. IUPRED is based on rather simple biophysical principles, and it calculates an interaction energy derived from how a sequence would interact in a globular structure. It's widely used, and it's also one of the earlier methods, so it's a nice case. where the total energy contribution of disorder has been estimated. The first assumption for a UPRET during the training is that you can take a set of structures from the PDB and you can just calculate the overall energy from an interaction energy matrix, which is derived by counting how often you see pairs of amino acids in contact. So these parameters are calculated from structural data, hence the PDB. And if you take this M matrix and you multiply it by the number of interactions of amino acid type I and J, you get an energy estimate. The underlying concept is that the more contacts you have for a certain residue in your structure, the more likely it is to be ordered and part of a globular domain, whereas disordered regions have few contacts. The same can be estimated just from the sequence. If you have a similar matrix, which you need to derive, which is called the P matrix, and you derive this matrix and you can estimate just from the sequence the energy you would get. So in this case, and I am glossing over all the details, but it's just to give you a feeling for the predictor. If this is the structure of the PDB structure 2KKW, this would be the contact energy derived from the PDB with one formula, and this would be the estimated energy from the sequence. And you see that the region which is less structured has a different energy than the one which is structured. So you can take this as an estimate for disorder propensity. Once again, you can see the eigenvectors of the P matrix to try to establish what are the factors involved in moving a sequence towards disorder. So the negative eigenvectors which are stabilizing are hydrophobicity and cysteine content, which is very, very much in agreement with the previous studies I showed you. Whereas the positive eigenvectors, there's the high net charge, the structure breaking amino acids, and then there's a different, and see here, a different contribution which has no clear explanation. But so again, you can see that IUPRET essentially captured the main features which were already established in the previous slides, except for the lack of conservation, which is not directly taken into account in IUPRET. Finally, we've been also contributing machine learning predictor in this field, which was in our case done by Ian Walsh, a postdoc in the lab who's shown here. And in the latest incarnation, it's called eSPRITZ. And this is a fast predictor built on neural networks, specifically bidirectional recursive neural networks, or BRNNs. And in this case, it was trained with 3 different data sets, one being X-ray short disorder, so missing residues from X-ray structures. The second is from disparate data, more about that in a few slides. And the third one from mobile regions in NMR. So the input of eSPRITZ, it can either be the single sequence, which is fast and slightly reduces overall accuracy, or it can be a PSI-BLAST profile. In this case, it's slower, but it is also more accurate. And this is a significant trade-off for the prediction of intrinsic disorder, because with this figure you can see here, on the x-axis you have the time required in seconds to compute predictions for 1% of the human genome. And notice that this is a logarithmic scale, whereas here you have the area under the receiver operator characteristic curve AUC. And notice that it ranges from around 0.83 to at most 0.9. So it's quite compressed in absolute terms. And here you can see that you have essentially 2 groups of methods. One, which are the methods using PSI-BLAST profiles, so using a full multiple sequence alignment, and these are slower by several orders of magnitude than a group of fast predictors, of which the eSPRITZ is also part. eSPRITZ S, yes, it's this one, which is 3 to 4 orders of magnitude faster. And in order to achieve the speed up, you're sacrificing only less than or at most 0.02% of overall AUC, which is quite negligible. In other words, you can have a method which is orders of magnitude faster than the ones requiring the full Psi-BLAST profile, and at the same time you sacrifice only very little in terms of overall accuracy, which may be advisable in many different for many different use cases. So here then, finally, to wrap up the part about predictors, one development I wanted to mention is FELS. And FELS is a fast estimator of local latent structure. Essentially, this is a web server where we bunch together all the different type of biophysical properties you can predict from the single sequence. They had to be based on single sequence, they had to be fast, and we also wanted fast predictors in order to try to get as broad a biophysical characterization of the single amino acid sequence as possible. So we have been using eSPRITS for disorder prediction, SAG for sequence complexity, a new secondary structure predictor called FES, which is also based on single sequence, then propensity for aggregation, hydrophobic clusters, charged clusters, amphipaticity for alpha helices and beta strand, and sequence composition. So the result of this, I will show you again for CDKN1B. So remember, we had this very nice structure from AlphaFold. And here you have on the left-hand side the prediction from FELS for all of the CDKN1B sequence, annotated in different various ways. And here in the middle, you have the crystal structure of CDKN1B bound to the cyclin CDK complex. And you can see that it essentially wraps around the structure here. There are some secondary structure elements highlighted here: helix 1A, helix 1B, strand 2, helix 3 and 4. And here you see the regions highlighted in the FELS results. Notice that these from the crystal structure correspond quite well with this region here, partially structured in AlphaFold. So this seems to be a case where AlphaFold captures the conditional folding of this disorder protein when it's bound to its binding partner. And here there's an interesting bit. So this region here is zoomed in on the top part, and you can see that you have a strong signal for alpha helix, which is here, this reddish color in this region. And this is exactly where the helix is then formed when the protein is bound. So this is the first analysis, which pits alpha helix and beta strand on the top side. So in this red purple color and this yellow gold color. And the height of the scale indicates the propensity to form that type of secondary structure. Below the central axis, you have a propensity to be in local coil structure. Then here you have in black the hydrophobic cluster, the presence of hydrophobic clusters, versus the presence. Oops, sorry. My connection was briefly jumping. Below in red, you have the propensity for disorder. And then you have here propensity for amphipaticity and other features. So essentially what this is telling you is that the single sequence already encodes a propensity to form the secondary structure, which is then also picked up by AlphaFold. And that this propensity for secondary structure is largely due to there being an amphipathic region. An amphipathic region means that you have interspersed amino acids, which one side of the helix is hydrophobic and the other side would be hydrophilic. However, there is also charged residues in this area. So the helix is not stable on its own, and it requires a binding partner in order to be stabilized. And this is interesting because all of these features you can derive from FELS in a few seconds, and they coincide very well with the full crystal structure and also with the propensity for structure you get from AlphaFold. So once again, AlphaFold seems to be catching the right signal in this area. So another thing I now wanted to address are the databases for intrinsic disorder, and specifically the disorder evidence from the literature. So here we have the DISPROT database, which was originally published in 2005 and was developed at that time by Keith Dunker at Indiana University in the US. And it has been growing for a number of years, but then it was in a stage where it was not being maintained so much. So starting in 2016, we took over the maintenance of the database, which was moved to the University of Padua and completely re-annotated. And then we started via a community curation effort to add new entries to DISPROT. And over a course of a few years, DISPROT has grown from being roughly 600 proteins to over 2,000. And this was largely the effort of a large group of different people, which are distributed in over a dozen different laboratories across Europe. So I'm very grateful to the over 60 different curators which have been contributing over the years to DISPROT, which have allowed us to grow the database. So what is DISPROT? Well, it's a manually curated resource of intrinsic disordered proteins. We just published the newest version of the database. And for those who are interested, there is a protocol about exploring manually created annotations of IDPs with DISPROT, which was published in 2020. And currently, DISPROT is largely maintained by Dr. Federica Quaglia as lead biocurator for the resource, who's hosted in our lab and who reviews many of the entries which are submitted to DISPROT by the community curators. So the DISPROT interface provides you, some basic statistics per organism. It now features also some thematic data sets. Here there's still only one. By now there are several on the web page. It also features its own blog and Twitter account for if you're interested in the news. But essentially, at its core, DISPROT has annotation about single proteins, and it shows these brief snippets here where you have the information about a region which is disordered. The information from these snippets is visualized in the feature viewer you see on top here. And this is a simple visualization, as you've probably seen also in the likes of Interpro or PFAM, where you have the equivalent of what in this case is not a globular domain, but rather a disordered region. Importantly, the various snippets which annotate the disordered regions are always taken from the literature. So you will always find a reference to a scientific publication. So you can go back and click on this link here and go to PubMed and see why this protein was thought by the authors of that paper to be disordered. There is usually a statement describing what has been seen and why it was thought to be disordered. And you have a header here which tells you the fragment of the protein, so which residues are disordered, what ontological term, but more about that in a few slides, has been used, and the experimental method which has been used in order to determine this as being disordered, as well as the curator for that region. DISPROT nowadays contains a large number of proteins, which are about 2,700. So, and here you see the growth over the different editions from DISPROT 7 to the current one. It has increased about, constantly, and by now it's a third, and a third what has been annotated. Of importance, for the latest release, there were another 940 proteins added and 2,800 and something protein regions. So these are always taken from the literature. Then the annotation in terms of species in this plot is shown here. And you can see that 3/4 are eukaryotes. And within the eukaryotes, metazoan proteins are predominant. There is, however, also a small chunk of viruses, which has been expanding recently because we have been deploying a new thematic data set for viruses. And of course, the human proteins are predominant because a lot of experiments are obviously done on human proteins, but you have also distribution of other organisms present. What this plot then does is it summarizes the information from the various annotated snippets in this type of consensus you see here. So this plot carries different subtypes of information about the structural state, the structural transition, interaction, and function. And these are compressed in the top line, the disparate consensus, which carries the various colors. So whenever you see something highlighted in a color in the disparate consensus, it means that there is evidence for disorder for that region. At the basis, it was annotated as disordered, but then there may be information about the structural transition, say from disorder to order, and there may also be information about an interaction between that region and another protein. So going back to our example from P27, the bound region would be highlighted in this form here. Now, the molecular functions of IDPs are an important piece of the picture, of course, because it's not sufficient to know that a bunch of residues are disordered. You will also like to know what they are doing. And here there are two basic classifications which had been originally proposed. The first one being by Peter Tompa from 2002, where he just came up with 5 broad terms for functions. And then Dunker and co-workers proposed 28 more detailed terms in this list here. And of course, when you have these definitions, you then want to bring them in line with other initiatives. And so we have been developing the disparate ontology based on this information, but expanding it and moving towards other established ontologies in order to improve the interoperability of the disparate resource. So at its core, we have 5 different aspects of disorder. Here you see the name and what it is about. The structural state is just whether it's disordered, ordered, or a multiglobule, for instance. And the ontology used in this case is the IDP ontology. Then you have the structural transition, whether it's disorder to order, order to disorder, etc. And again, the IDPO. Now, in the latest release of this plot for the interaction partner and the disorder function proper, we have been collaborating with gene ontology, so that we are very happy that several terms in the gene ontology have been updated or expanded in order to cover different types of functions which are specific for disorder proteins. And then there is a residual part of terms which are only in the IDP ontology for reasons I'll explain in a minute. Last but not least, there are experimental methods of how the various proteins have been thought to be disordered, so how this has been proven. And here we now use ECO, the evidence and conclusion ontology, which was also expanded to cover these instruments. Let's start with ECO, the evidence and conclusion ontology. So this contains terms, classes that describes types of evidence and assertion methods. And this has been expanded in order to cover the different types of experimental information you can have and different types of experiments you can carry out for IDPs. So as one example here, you can see a small angle X-ray scattering evidence, which has this definition, a type of light scattering evidence derived from determining the properties of particles in a solution, such as size, shape, structure, etc. And it's fully integrated into the ECO hierarchy. So the collaboration we had with ECO has been very fruitful, because now all of the experimental methods which are present in this plot are also included in ECO, which has been expanded, and therefore this allows much broader interoperability of the experimental evidence from Disprot. But also Disprot has been looking at different types of functions, and we are currently using all of the so-called active functions and through the gene ontology terms, which have been expanded, updated. This is another major step because you will soon be able to find the Disprot gene ontology terms also in other databases because they will be propagated. There are, however, a set of passive and self functions which do not fit well into the gene ontology schema. And therefore, these are so far left outside of gene ontology. We are in conversations with the gene ontology consortium about sooner or later bring these also into gene ontology, but for the time being, these are outside, and these are essentially entropic chain, self-regulatory activity, and display site. So the gene ontology, you're probably familiar with this, is a way to describe with ontological terms the function of proteins in three ways: molecular function, cellular component, and biological process. So the functions of IDPs have been included mostly in the molecular function and a bit in the biological process, so that you now find additional terms which were previously not available. And these are already in the current gene ontology setup. The so-called passive and self functions of disorder, which are specific to Disprot and the IDPO and are not currently in the gene ontology, are of a different kind, because gene ontology only covers functions which are carried out by the protein. But here we are talking about things which are the self-inhibition or self-activation of the protein, so that the protein, depending on outside circumstances, such as a binding partner or PTM, for instance, starts to change conformation and therefore self-inhibit or self-activate itself. And this can be a different, something which is not explainable per se in gene ontology. So you find the terms only in the IDP ontology. And also the entropic chains, which are essentially flexible linkers and different variants of this. These are difficult to represent in gene ontology, hence you can find them only in Disprot and not in the gene ontology. Here you have an example of one protein from Disprot, where the statement is that there was a disordered 15 amino acid linker not visible in the electron density map, which is here. So this region of the protein would then be annotated as disordered because you have a crystal structure, but this was missing, and therefore it is considered an entropic chain, specifically a flexible linker. So with this, I think we have time for a break, and I think we can probably reconvene at 12 sharp after a short break and bio. Yeah.
Okay, thank you. Is there some question for now? Otherwise, we get a break.
I think we can probably do all the questions at the end.
Okay, see you in 5 minutes.
Okay, thank you. Okay, so so far I've been telling you about the predictors of disorder and the main database for experimentally QDated evidence. However, there's more you can do in order to try to annotate disordered proteins. And this is demonstrated by the MobiDB database, which is an attempt to map and combine different sources of IDP data. So specifically, MobiDB operates on a data quality versus coverage pyramid, which contains at least 3 different layers. So at the top of the pyramid, there are curated databases which contain information. This is largely Disprot, but there are also a number of other databases which contain information. For instance, UniProt contains some proteins which are annotated as disordered, although they are very few of them. Ideal is a database similar to Disprot, and then there are other databases. Elm is for short linear motifs, and some smaller databases, which can all be considered curated data and form the tip of the iceberg here. So the advantage of these is that they are high quality. There is curators who verify that this is experimentally disordered information, but there's only so many of them. Below this, you have a layer where you can use derived data, which is essentially extracted from the protein data bank. So the regions of crystal structures which are missing residues can be considered a proxy for disorder. Of course, this may not be the real thing. There may be many different reasons why they are missing from the protein data bank, but still it is indicative that something is going on. Likewise, you can also use information about NMR structures, the parts where they are particularly flexible, to get a proxy for disorder. And this would be the derived layer in MobiDB. In this case, obviously, there's more information available. We're talking about, for databases, the order is a few thousand entries. For derived data, we are on the order of 30,000+ different proteins which are annotated. And below that, you can have predictions. So predictions you can run in principle for all of UniProt. And this is done through the software called MobiDB Lite, which I'll be describing later on. But here, essentially, in order to run it on all of the UniProt sequences, which now count over 200 million proteins, you really need only a fast method. This is why it is important that there are fast methods available for predicting disorder, because otherwise it would not be possible to cover this. So this is a more technical view of what MobiDB does. It has its different input layers. This is the database again with its various components. There's many different pieces of software and data sources which are being integrated. Currently, we have about 200 million sequences which are annotated, and these are exposed both through the MobiDB website for the human user, as well as through the MobiDB API for computational and programmatic access to the data, and also through MobiDB Lite as a piece of software which is can be installed locally. This has been used by various types of third parties, and I encourage you to visit the MobiDB webpage in order to see what it does. So the curated data, this is the tip of the iceberg, as I was mentioning, is essentially Disprot and a few other databases. There may be conflicting annotation here and there, and this gets put into a consensus prediction. So again, you have the feature viewer, which summarizes the information. And here you have an example. So in this case, you have regions which are always, annotated as structured, regions which are always annotated as disordered, and regions which are partially annotated as structured, but also as disordered, and therefore they become these conflicting regions. So as a shorthand, these conflicting regions can be parts where you have disorder to order transitions upon binding. The second type of information is derived from the PDB. I already mentioned missing residues or high temperature factors, mobile residues from NMR models. And once again, these go into the feature viewer, and we have these three classes: disorder, order, and conflict. Predictions are calculated for all UniProt KB sequences, and the consensus is calculated based on the agreement across predictors, and shown here as a two-state representation, or in a more continuous fashion, it's shown here for MobiDB Lite. There's also a length cutoff, so that for the MobiDB Lite regions annotated, you don't get many very short fragments, but rather you get the more interesting and more relevant longer disordered regions. Here you see an example of the MobiDB simple view for P53, which is a well-known protein. So you have in the top part the basic description of the protein with information fetched also from UniProt and its interactors. This feature viewer here is really the core of the information you get from MobiDB, and below it you have cross-references to other databases. This is the simple view, because by clicking on this button here, Advanced, you can open up the view to something much more extensive. So now you get the full lot of information, of annotations from MobiDB, where you can get the consensus with its various subtypes, information from the PDB, curated information, but also predictions and various types of other predictions as well. And here you have an overview when there are protein structures where a fragment of your protein is bound to something else. This is shown here. This is the structure, and this is the residue contact network. So you can get a feeling from these representations here whether you're looking at a short linear motif, which is bound to a globular domain, for instance, which may be useful in order to determine the function of different disordered proteins. So MobiDB has as a goal the integration of different sources of information for IDPs. And once again, through Elixir, we have been able to do much in terms of integration and standardization of IDP data. So at the start of this whole endeavor, IDP annotations were significantly underrepresented in Elixir core data resources. These are the really large databases such as UniProt or Interpro, for instance. And the goal here was to integrate IDP data resource to provide a blueprint for integration of further IDP data into the CDRs. And the MobiDB Lite tool has been used for the automatic structural prediction of IDPs. So this was the status when we began the work. This is a Venn diagram showing the overlap between different databases which are providing annotation for disorder. And you see that there is essentially nothing in the core. Even at that time, Disprot was already the larger of these databases, but it had very little overlap with most of the other databases. So integrating the data was important. And here you see a different view where you have the overlap between Disprot annotations, Ideal, which is another database from Japan, similar to Disprot, and the long disordered regions, which you can find in the Protein Data Bank. And you see again that there's very little overlap, but that the Protein Data Bank can actually provide you with a lot of regions of missing residues, for instance, which are by now also quite long. And most of them, if not all, are in fact disordered. Hence, the MobiDB Lite tool, which we have been developing, is based on the assumption that you would like to have something which is essentially the opposite of a globular domain. So a globular domain has a certain length threshold and it folds in a given way. So in this case, we are looking for longer disordered regions. The cutoff is set at 20 amino acids. So anything above of at least 20 consecutive amino acids, which are reliably predicted by a majority of methods to be intrinsically disordered. And this is therefore something which can carry out a function while it is intrinsically disordered. So in order to come up with this predictor, we analyzed various different predictors we have in MobiDB. And this is about 10 different predictors. And on average, here you see plotted per protein. These are the different types of predictors. So this would be MobiDB Lite itself. This is the sample hot loops. This is VSL, which is a fast predictor, globplot, eSpritz for NMR, IUpret short, and some other predictors. Each one of them, taken alone, has a significant amount of overprediction for disorder. This is because it's difficult to recognize exactly where the disordered regions are, but many predictors recognize that there is disorder. So there is overprediction for several methods, and if you combine them, you come up with MobiDB Lite, which is a majority consensus. And in this case, you have very little overprediction because we are using a very stringent definition. This comes at the cost, however, of having a somewhat larger degree of underprediction. Down here in blue, you see the fraction of underpredicted disorder. And you see that, of course, the predictors which say that many residues are disordered also get get fewer problems of underprediction, and vice versa. The ones which are more conservative here tend to predict a bit more. Tend to miss out more regions as being disordered. So the end of the story is that MobiDB Lite is a very conservative predictor, which has this length cutoff of 20 residues. And whenever it detects something as being disordered, it is extremely likely. In the benchmarking set, it was over 95% probability of being disordered. But this comes at the cost of missing out certain regions which may be disordered and are not picked up by the method. But again, this is intentional because it was supposed to be the opposite of globular domains. This highly conservative predictor has allowed us to migrate the data from MobiDB into various core data resources at the EBI. Specifically, MobiDB Lite is integrated in Interpro, so that now Interpro, for the last few years, rather than giving you only the globular domains, also gives you information about disordered regions. And this is the Interpro output as you can get it. Here under other features, you have the MobiDB Lite consensus, and this region here is listed as disordered. So if you're interested in the annotation, then you can click on this region and go back to the MobiDB web page. Once the information is stored inside the EBI ecosystem, it can be migrated from Interpro to Uniprot and the PDB Europe. So by now, the MobiDB Lite annotations are used by Uniprot, for instance, to automatically annotate disordered regions via sequence similarity. And they are also shown in the Protein Data Bank Europe as annotations for the protein structures. Here you see an example where this is indeed called flexibility, as Rita likes to call it also. So this has taken us further, and at the moment, what we are working on is to be able to integrate the information from the various distributed resources which are dealing with the manual curation of disorder into a unique platform or portal, if you like, which will be called IDP Central. So the collaboration between MobiDB and the EBI resources has been the blueprint for this. And the idea here is to have IDP Central as the portal for the data which will then be brought into the Elixir core data resources, so that effectively the definitions of what is thought to be disordered will be coming from IDP Central. And here we will be integrating not just databases which curate the literature for disordered regions, such as Disprot, but also other databases which are providing primary data, for instance, for SAX data, which is SASBDB, where the SAX data is deposited. We have a collaboration ongoing with them. The PET database for protein ensembles is by now also located physically in Padua, but it's an ongoing collaboration with Brussels. The circular dichroism database PCDDB is also being leveraged for having the annotation more easily integrated, and BMRB, which is the main repository for NMR data, is also part of this effort. And the standardization, which is necessary in order to put all of this data into one place for IDP Central, is carried out through the HuPOPSI ID working group. So HuPOPSI is the Human Proteome Organization Proteomic Standards Initiative, and it's the IDP working group. So this is the main player in the field of standardizing data related to proteins in the widest sense. So there is an ongoing standardization effort in this area, and that will allow much more interoperability between resources, so that this will become easier. So far, so good about all the databases and predictors for IDPs, but now I will be telling you about a critical assessment we have been carrying out for disorder predictors. And the last part of the slides will also show you how this relates back to AlphaFold. So to answer our question about the weird protein with the ribbons we've seen in the beginning, and whether those regions are disordered or not. So I think in the introduction, Rita mentioned that we are eager participants in various types of critical assessments over the years. And so since this intrinsic disorder had not been benchmarked for a decade, because the CASP experiment stopped benchmarking it around 10 years ago, we came up with our own critical assessment exercise, which has been published last year in Nature Methods, and it's called the Critical Assessment of Protein Intrinsic Disorder Prediction, or KIDE. By the way, there is the full data set attached to this paper available. Here are the links. And there's even a code ocean capsule if you want to rerun the full evaluation. And we have tons of supplementary tables and figures which can highlight the various aspects. So regarding the assessment of IDP predictors, this was originally carried out by the CASP experiment between 2002 and 2012, so CASP 5 to CASP 10, because originally it came out that there were missing residues which could be considered intrinsically disordered. So this was used as the mainstay in CASP for defining which parts of the proteins were supposed to be disordered. In the later stages of the CASP assessment, mobile residues, part of NMR targets, were also seen as disordered, and there was a manual curation process, in particular by Joel Sussman's group, in order to identify the targets which could be considered disordered. So at the end of the day, the CASP experiment was run on relatively few targets, only about 100 of them, and there was a prevalence 7-to-1 of short over long disordered regions. In the whole data set, only about 10% of the residues were considered intrinsically disordered, so it was a rather marginal data set. Moreover, there was a strong bias towards region at the N and C termini, because essentially the CASP targets were proteins which had been either crystallized or solved by NMR, so they were by definition predominantly structured. And this had its limitations, and therefore at the end, in CASP 10, the organizers of CASP decided that they would discontinue this category because there was not enough good data in order to do the assessment. And after this, of course, with this prot, we have been able to annotate a lot of additional regions for functional disorder via the so-called secondary methods, which are all those different types of indirect information, be that CD spectra, SAX data, FRET, or other techniques. And so the new DISPROT from 2020 had a whopping 764 proteins which were entirely new, and they hadn't been seen by any of the predictors. So keep in mind that this was data which was completely blind for the predictors because they were trained on their own data sets, and these were simply not available in DISPROT at the time when the predictors were developed. So out of these, 646 non-redundant proteins were used for the first round of KIDE. And this puts us in a situation which is as close as possible to what the CASP predictors usually do. Moreover, with the whole infrastructure we had in place, so on one hand, the Disprot database for curating IDP information from the literature, which guarantees that there are papers supporting the fact that these proteins are disordered, and the standalone software which was installed on the MobiDB servers, and therefore we already had a bunch of predictors locally installed. The idea for KIDE is simply put to run through a cycle where you do you annotate proteins in Disprot, you run the predictions on the MobiDB servers, you do the evaluation, and you report back the results of this, and this feeds into the next cycle of KIDE. So the KIDE 1 experiment was mainly run by these two people here, Dr. Damiano Piovesan, who's also the project lead for the entire KIDE experiment, and Dr. Marco Necci, who was providing most of the analysis at that time. And the basic concept of KIDE is that unlike CASP, where the predictors take the information over the web, they download, they see the sequences, and they do whatever they like on their own computers and submit the results. With KIDE, we wanted to install the software locally. And this is a design decision because we believe that for disorder prediction to be useful to the experimental community, it should be a piece of software which you can install locally, and it has to be reasonably fast. Of course, this excludes a number of pieces of predictors which are only available as web servers, but this was a design choice, and we stand by this decision, and we believe that it has served us well for the overall KIDE experiment. Nevertheless, keep this in mind. So the assessment in KIDE then is carried out both in terms of accuracy and in terms of technical evaluation. And the technical evaluation is again something which differs from CASP because we have the software installed locally, and so we can say things about the execution speed on our machines, for instance, and we can compare the methods. So the prediction setting is fairly straightforward. As input to the software, we give them a FASTA formatted sequence file. So this is the plain protein sequence. And optionally, we can provide also a position-specific scoring matrix from SciBlast or a multiple sequence alignment. This is fed into the black box, which is the predictor itself. And out of the predictor, the black box, we get the prediction, which is a two-state thing. So residues which are thought to be ordered, residues which are thought to be disordered, and we can also have a score attached to each single of these amino acids, and this will be recorded. So it's based on standalone software. It had to be installed on the local machines in our lab at the University of Padua. So the requirements are that it is a SunGrid engine cluster with nodes have different hardware, but no GPU. It is a Ubuntu-based Linux distribution, 64-bit, with 16 gigabyte of RAM and 8 cores per node. This was the setting. And we received in total 27 software packages for evaluation. 8 of these were unpublished, and 5 of these were recently published at the time we collected the predictors, which was in 2017. So this in total produced 43 different types of outputs, 32 for plain disorder prediction, and 12 for disordered binding region prediction. So these are subsets where the predictor thinks that those residues are involved in binding to something else, which may be a protein, DNA or RNA, or another molecule. We had many different issues when installing the software, and this has been a valuable lesson for us. So first of all, there was many different types of output formats. Some of the software were large. In particular, one group submitted 60 gigabyte of data and code in order to run the software. There was no control on output paths. Some of the software required specific versions or obsolete system libraries. On the contrary, some of the software compiled only on new hardware. For instance, they were using TensorFlow, and only one was provided as a container or virtual machine. So this is one of the suggestions we make for the community now, to provide, if possible, the software as a container, so that it makes local installation easier. Regarding the no control on output paths, there's also an interesting story where one of the predictors actually behaved like a virus, because in the script to launch it, it would just remove everything which was in the directory it was launched on. So it was an RM wildcard. And fortunately, this has been carried out in a subdirectory and not damage anything else. But this is just to give you an idea of how there are also technical issues with the prediction software, which are in many cases not fully thought about by the authors. So now the important bit here is the definition of the ground truth for KIDE. So of course we use the disparate regions as annotation for disorder. And here we have two different types of regions. The ones which are just annotated as disordered in disparate, shown here in red, and the ones which in disparate are shown as disordered and also as binding some other molecule. So we were not distinguishing between types of molecules in the first round of KIDE, and we probably will be doing that in the second round, because most of the disordered binding regions were actually due to protein binding, protein binding, but at that time we didn't distinguish. And then regarding the structured parts, so the opposite of disorder, you can take two different views. Either you can look at the protein data bank, where you have X-ray structures and NMR structures with coordinates. These would be shown here in blue. And then the two references you can get out of this is, on one hand, the disparate definition, which says disparate disorder stays for disorder. Everything which is not disordered in disparate is thought of as structured, shown in blue here. And the alternative view would be that you take the disparate disordered as disordered, but you consider only the parts which are structured in the PDB as structured, shown here in blue. In this case, you are throwing out larger chunks of the reference sequences, shown here in white. And this reduces the data set quite a bit. So it is a design choice. And both of these are valid, and we will see the results in a minute. Suffice to say that most predictors, when they benchmark the method, they do it on the disparate PDB definition, because it's also easier for the predictors. You get nicer numbers, you get higher AUC values, for instance, if you run disparate PDB than if you run the disparate definition. Last but not least, we also have the disparate binding definition, and in this case, we just say that disparate binding is positive, the positive case, and everything else is negative. Notice that that this definition is probably missing some other potential binding regions, and this is more similar to what you would have in Kappa, where also for function annotation, it may be that there are other functions and that they are just not annotated. So likewise, in disparate binding, there may be regions which are binding, but they hadn't been annotated. And so it's a sort of open world problem. Now, if we look at the data set composition, if we take the disparate definition, which is wider, then we get the whole data set around 350,000 residues and of those, something like 70,000 or thereabouts are disordered. The rest is ordered. If you take the disparate PDB definition, the amount of disorder stays the same, but the amount of structured residues is roughly halved. And in total, the data set is much smaller, and this has to be taken into account. For disparate binding, the amount of residues thought to be disordered is about 30,000 or thereabouts. So in terms of proteins, here you see the Venn diagram. And interestingly, there is a subset of proteins for which there is annotation in disprot, but not in the PDB. And these 74 plus 65 would be completely lost in the disprot PDB definition. This is a further argument why disprot PDB is perhaps not such a good idea as a reference set. In terms of taxon distribution, it's pretty much like overall dispro, so I've already talked about that. Moreover, we've added some baselines used for comparison. And in this case, we use the PDB information, be this observed, so 100% identity with the PDB close relatives or remote relatives, with a 30% identity cutoff to PDB found via PsyBlast. Gene3D gives you HMM models representing the CAF domains, so it should be somewhat more expansive than the PDB remote baseline. And then we have two simple baselines for comparison: the shuffled data set, which just randomly assigns residues as positive or negative by keeping the ratio at the data set level, and completely random, which is just a random reshuffling. So let's see, first of all, the prediction success and CPU times for the top 10 disorder predictors using the disprot data set. In this case, what you see is that the Fmax values are lower. The top predictors score less than 0.5. So specifically, it's 0.48, and they have 100% coverage. So these graphics are similar to what you would get in CAFA, for instance. And this is the precision versus recall curve. And you see that the top predictor in red goes like this. So it has a peak at high precision at low recall, and then it goes down. You have other predictors which are performing slightly worse than this, and the baselines are shown as these various symbols down here. So first of all, the good news is that the top predictors perform better than the baselines. So there is still use for all the predictors which are being developed. It would be rather awkward if this were not the case. But nevertheless, it's reassuring to know. Then the second thing is that down here, you see the time in seconds for executing all of these predictors. And notice that this is a logarithmic scale. So from 1 second all the way to 10 to the 4th seconds. And you can see that there is quite a bit of variability in the top performers. So there are some methods which are 1 or 2 or even 3 orders of magnitude faster than some of the top performers. And yet there is relatively little difference in terms of overall quality. Of notice, FIDP-NN and FIDP-LR, which are by the same group, which is Lukas Kurgan's group, are faster than the other top performing methods, except the ones coming way later. And so there is a good margin. And for this particular data set, FIDP-NN and FIDP-LR stand out as a good compromise. If you then look at the disparate PDB data set, so keep in mind, this is the case when you throw out all the residues which are not structured in the PDB and not disordered in disparate. So we are roughly halving the overall data set. Here we have a surprise. So there is some reshuffling of the top performing methods. FIDP-NN is no longer at the real top. It is the 10th out of these 10 methods. There are others performing better. Some of the methods which were doing well before, so spot disorder, for instance, have slightly improved. They tend to be of the smaller kind, although there are some fast methods down here which perform quite well. But the biggest surprise really is the performance of the PDB observed. In this case, PDB observed would be this point over here, and it is clearly better than all of the predictors. However, there's a catch to this, because PDB observed by definition is only what we have been evaluating. Remember that we're taking as negatives for disordered only the regions from the PDB. Hence, by definition, this is considering as structured whatever is in the PDB and as not structured whatever is not in the PDB. And so it happens to be an overly expansive baseline. However, this also tells you something, that the PDB is a valuable source of information for disorder, and that whenever you have information about structures in the PDB, it's well worth to use it. Besides this, the other baselines, some of them still outperform some of the predictors. So these are the PDB observed and the PDB close and gene 3D. Because again, if you know what is all that is structured in the current PDB, you get a good approximation of what could be disorder. However, arguably, these methods are not annotating for disorder. They are really annotating for things which are unresolved in the structures, and these tend to be things which don't crystallize. So arguably, they are not geared towards predicting disorder, but rather they are picking up signals for the parts which will never be crystallized. This may sound like a subtle difference, but it makes quite an impact if you think about it across all of the proteome, because there are regions in the disordered proteins which probably don't have the hallmark of what is being picked up here, which is essentially the opposite of structured domains, but yet they are disordered. So to cut a long story short, this is a different baseline. This is a different reference. It gives a stronger impact for the baselines. The predictors like it because their numbers are much higher. The Fmax here is around 0.8. Contrast that with 0.48 for stricter disprot definition. But it also makes the predictor somewhat redundant. So for the future, in KIDE, we will be sticking to the disprot definition because we believe that there are a number of arguments why this should be preferred. Then one of the types of analysis we did for KIDE, which was not possible in CAS previously, is that we had a subset of fully disordered proteins. In this case, these were 41 versus 603 proteins which were just disordered. So there was some disordered in it, but not too much. And here, the various predictors, well, they are somewhat reassuring in the sense that, for instance, FIDP-NN picks up most of the proteins which are not fully disordered as true negatives. It has around half the data set as true positives, so it picks up half of the fully disordered proteins as being such, and then it has some errors in the middle. So this means that the Matthews correlation coefficient for this problem is still quite good. Here you can see the performance of PDB observed. This is another argument why you should not just rely on what is in the PDB as saying that everything else is disordered, because you get a lot of false positives in this case, 133. So overall, it's fair to say that the single predictors, while they may not always be able to pick up the fully disordered proteins, they can distinguish them fairly well from most of the structured ones or of the only partially disordered ones. Still, this is a subset of data which is interesting, and we'll be looking at it in future rounds of KITE. Then in terms of prediction, success and CPU times for the top 10 binding predictors, in disparate binding, there are a set of mostly biophysical methods in order which try to predict this type of feature. The top performer here is Anchor2. Anchor is by the same group, Shoshana Bustani, which developed also IUPRED, and it uses a similar concept. The F-score in this case is 0.23, and reassuringly, it performs better than all of the baselines, which is good. So the top performers in this area are really contributing something to the scientific question, which is always very good. And there is, again, a wide variety of computational times. Two important caveats is that the number of binding regions are probably unknown, and that disparate annotates longer regions compared to the actual binding positions. So there may be some data set issues on this side as well. This is an interesting part, which is still undergoing development of various methods. And so we're looking forward to the next round of KITE to see if there is any improvement. So some conclusions about KITE. The novel predictors tend to outperform the old ones. Evolutionary information is useful. Disorder is different from missing residues, and binding regions are difficult to predict. A number of predictors are difficult to use, and disparate as a ground truth is better than the PDB, missing residues. So overall, it was an insightful experiment, and we are very happy about all the work which went into this. Once again, this was a community curated experiment. So this is a list of disparate curators which participated in the data set. This is the group of predictors who have been contributing their software for the KITE evaluation. And I already mentioned the two people which have been contributing to this. So now we're about to start a new round of KITE this year. In fact, the software submission deadline at the moment is set at the 1st of April, 2022. We have an agreement in place with the CASP organizers that the final results of this round of KITE will be announced at the CASP conference to be held this December. And there may be some extension to the software submission, but our main goal is to have the software successfully installed in May, and to be able to have preliminary results in July, so that we have a stable set of results which we can then announce at the CASP conference, and the publication of the results in 2023. Now, of course, you may be asking yourself, what about AlphaFold? Well, in fact, we've also been asking ourselves, what about AlphaFold? And remember that there is this rather large fraction of amino acids in the human proteome which are not well annotated by AlphaFold. Well, you can use the two different ways to have a proxy for disorder in AlphaFold. The first one are the low PLDDT regions. So these are the ones predicted with a low quality score by AlphaFold itself. And this seems to correlate very well with intrinsic disorder. But also you can look at the regions which have high relative accessible surface area, which also tends to correlate well with intrinsic disorder. Or finally, you can make a clever choice between the difference of PLDDT and RSA, and you can try to see whether you can recognize regions of conditional folding inside an IDR. Well, if you look at an example from our disparate data set, which we had part of KITE, and here you see the AlphaFold2 prediction for this. You see the numbers of the residues. This is the N terminus, this is the C terminus. So you see a helix, then a globular domain, a bit of helix, one of these long ribbons, another helix, and then something which is largely disordered with perhaps just a hint of some local structure. This is the overlap between the disparate annotation where you have disorder at the end from residue 253 onwards, which you see happens to coincide here with a region which has lower PLDDT score in AlphaFold. The PDB has this region between residue 25 and 167 structure here. Then Interpro annotates the same, roughly the same region as a globular domain, recognizes region from 1 to 25. So this helix here in Interpro is annotated as a signal peptide. And the region 225 to 250, roughly this region here, is a transmembrane helix. In other words, you would say, more or less, boom, bingo. AlphaFold got it everything correct in recognizing pieces which have an structural explanation and pieces which are probably intrinsically disordered. So after optimizing the score, and I won't go into the gory details of this, these are three versions we have on our hands on how you can use the AlphaFold scores in order to annotate for disorder. One is based on PLDDT, one is based on relative solvent accessibility, and one is a mashup of those two scores in order to come up with a binding propensity. And notice that the red regions for disorder match quite well, the C-terminal one definitely with the annotation from this plot. There's this other region here between residues roughly 165 and 230, which is not annotated as disordered in this plot. This may be a false negative from this plot. We are looking into this. And then the mashup, in order to recognize binding regions, provides this yellow annotation here, which correctly predicts this bit over here, but it also predicts some other bits and pieces across the sequence, as well as the N-terminus. Now, arguably, the N-terminus, this being a signal peptide, can be considered a bona fide disordered binding region because signal peptides are cleaved off and they serve in order to transport the protein by binding to another protein. And there is a bit of confusion on this yellow bits over here. This is the continuous score from AlphaFold. So bringing this to bear on the entire KIDE evaluation, here you see with, the full annotation for disprot PDB, disprot, and the binding regions. The red arrows point to where AlphaFold would be in these results, and you see that for the disprot PDB definition, it scores a top spot. This is easy to explain because remember that this is when we use only the regions which are available in disprot, only the regions which are available in the PDB and structured as negatives. And of course, AlphaFold excels on those. If you take the more realistic reference of using just disprot, AlphaFold can still perform very well. It's in the top 10. It's around position number 5 or thereabouts. But reassuringly, for all the disorder predictors, it's not yet the top performing method. And last but not least, if you use our mashup between the two scores, you can actually also use AlphaFold to predict binding regions. And arguably, these are the conditional binding regions where you have a higher AlphaFold predicted score because there is some local structure, but they are embedded in a region which is highly solvent exposed and therefore likely being unstructured. So the paper describing this is currently in preparation and we will be submitting it soon as a preprint to bioRxiv. So watch the space for more information. And with that, I would like to acknowledge all the people in my group who have been working on this and on other projects. There's quite a few of us. We also acknowledge gratefully funding from Elixir and from other initiatives, most notably IDP Fund, which is Marie Curie Staff Exchange Program, dedicated to intrinsically disordered proteins. And finally, I would like to leave you with one image of how this series of Bologna winter schools all started back in 1999. So thank you very much for your attention.
Thank you. Thank you, Silvio, for the lecture. Very exhaustive on the topic. And I ask the audience if there are questions for you. You can raise your hand or type in the chat window. There is a question from Michael Enfinger. Thanks for the great overview. Will KITE2 support GPU-based methods?
That's a very good question. So, well, first of all, we only need to run the executable for the prediction. So the training, if it's done with GPUs, we don't see it. So we just need something which is executed. In principle, we could be supporting GPU-based methods if there is a necessity to do this. So if there is at least one method which works only on a GPU-based system, we can talk about this. We have some resources for this. But once again, as basic philosophy here, I recommend that the prediction software should be possible to run on a standard Linux box, because that is most likely what the experimentalists will be using if they use local installations at all. So yes, in principle it's possible, but we would prefer to have a standard CPU-based executable. Okay.
Thank you. More questions?
So while there are questions, this was myself at that first summer school back in '99.
Okay, so the way we were. Listen to me, please. If anybody is asking a question, I will do myself. Okay, so you are perhaps much more aware than I am of these fashionable stories of the biomolecular condensates cross scales in literature, with several experimental evidences of these membraneless organelles that are obviously even time dependent, and so it's very difficult to follow them even experimentally. Did you ever try to build up such of these multi-scale condensates, adopting the proteins that are supposed to interact?
Well, there are several parts to this question. Regions undergoing liquid-liquid phase separation are being actively annotated in this prot. So there is a data set about that already available. There are also some other databases which are specific for liquid-liquid phase separation. One we've been collaborating with is called PhasePro, but there are several others which are large-scale efforts. So the basic annotation is there. We would love to run predictors for liquid-liquid phase separation in the next round of KITE. But so far, there's only very few of them, and it's difficult to get our hands on these. And regarding the actual atomistic simulation of the regions undergoing phase separation, no, we haven't been doing that. I'm also not aware of any specific attempt to do that systematically. There is some anecdotal evidence of people have been looking at one or two cases at the time, but the large-scale effort is still missing.
Well, nice idea to explore in the future.
Yes,
when I will ask you for a PhD position, okay? Okay, thank you very much for this brilliant lecture.
Thanks.
Then other questions around my students, the students of the master. Where are they? Are they there, or did
I think we have a question from Maria Giulia Prado.
Okay, but she's not a student of a master, a student in the master, as far as I know.
So, Maria Giulia.
Okay, can I, if I can talk?
Yes.
Yes, please.
Okay, so thank you very much for this very interesting lecture. I was fascinated when you mentioned before that it is possible to have an insight of the secondary structure that an intrinsically disordered region will fall to when interacting with a binding partner. So my somewhat utopistical question is, will we ever be able, in your opinion, to will we ever be possible to have an insight of which interactions or which interactor is needed for a protein to enter a disorder toward the transition? So sort of with very pharmacophorical map of the interaction that are needed to the protein to fold to have a second ordered secondary structure. I don't know if I explain myself, but
yes. So in principle, what we are looking at here is, well, let me just go back to the slide about felds. What this is telling you is that the, where is it? Here.
Yes, this was the slide I was referring to.
Behavior of the protein in isolation is largely encoded by its sequence. So there is an evolutionary optimization process whereby in order to optimally bind to its binding partner, the sequence is selected in a way that this binding affinity is increased. Of course, also the other binding partner undergoes a similar selection. So from the sequence itself, you can hope to recognize the presence of propensity for secondary structure, alpha helix, beta strand. What you can do in order to try to guess the interaction partners, and this is in principle what AlphaFold also does. There are extensions to AlphaFold where they look at protein complexes, that there is a sort of mutual information between the two sequences which are co-evolving. So the protein sequence, it has to evolve in a way that it is specific for its binding partner and at the same time doesn't bind to other partners. So there is a sort of evolutionary arms race that it only binds to the right ones by tweaking its sequence. You can think of the sequence in human proteins in a way as highly optimized for its function. And the differences you can have between amino acids in disordered regions, they can be seen also either as purely random, and there is a degree to that. If part of the sequence is simply not necessary, then it will be free to evolve. This is the concept shown here, these ones. So the non-conserved disorder. And these you can just add or throw away, and there's not much selection there. The flexible disorder tends to be the regions which there has to be something in there, but it's important that it keeps more or less the length. And in those areas, there already seems to be some conformational selection towards the better fitting residues. The constraint disorder, this one you see largely in the ribosomes, for instance, where you have partially folded protein chains, which are highly optimized in order to fit in there, and they are conserved throughout all of the ribosomes back to the first living organisms. So coming back here, well, the hope is that methods such as AlphaFold will be able to pick up those signals in order to give you an idea about the binding partner to be predicted. Other than that, there's the whole bunch of regular protein-protein interaction predictors. Yeah.
Okay, thank you very much.
Welcome.
There is a question in the chat by Maria Paola Turina. Do you think that it would be promising to devise by DeepMind people an AlphaFold2-like procedure in which IDRs are the target of prediction rather than a sort of byproduct?
Yes, I definitely believe that. And in fact, we we are trying to get them also to submit a predictor for KIDE. And of course, and this is also why the next KIDE round will be advertised with a CASP crowd, because there are several groups which can be developing things. Except for DeepMind itself, many of the CASP participants have also their own disorder predictor. So some of the usual suspects are already there. It's just a matter of onboarding the others. What you can also see from this type of result, so just contrast the amount of calculation which is necessary to produce this model here by AlphaFold versus what Fels does in 1 or a couple of seconds. So the signal really is already there. And ultimately, if you look closely enough at the protein sequence, you also realize why alpha helices or beta strands are being formed, because they have an amphipathic pattern. So that ultimately drives the secondary structure prediction and gives you propensity for that. So I'm actually, and this is without taking into consideration evolutionary conservation, so multiple sequence alignment. So I'm fully convinced that you can build some very good predictors which exploit all of this knowledge and are competitive or even better than AlphaFold if you just pick the right machine learning methods which are currently available. Okay.
So, more questions? If not, I thank Silvio very much for the lecture, and I remind you that the next lecture is tomorrow morning, Professor Orengo, and see you tomorrow. Bye. Thank you.
Thank you. Bye-bye.
Bye.
Bye-bye.